Hi everyone, welcome back to the next part of the NEET's 2025 question paper discussion for the biology. So it is a part 2. So let us discuss about the questions after the 120, because in the part 1 we have seen about the question number 122 up to the 120, and then after that we will see into this part 2 video. Okay, so let us discuss about the question number 121. Okay, so here it is a question number 121. It is telling it is asking for the matching. Okay, so matching for the centromere, cilium, crista, as well as the cell membrane. We all knows that phospholipids are present into the cell membrane. Okay, so that is a PLP, or the phospholipid. For example, lecithin we have seen, so D should be 4, and crista is a part of the mitochondria. Okay, so it is like into the mitochondria inner membrane. So C should be first. Okay, so C1 given in the first in the one option only, that is the option number 4. Next is the question number 122. That is again matching chlorophyll A. Will like chlorophyll A is actually having the blue algae. Okay, so we can say like chlorophyll A is actually the part in which like blue algae should be matched. Okay, so it should be A3 given here and here. Okay, and the chlorophyll B would be the part like the yellow green. Okay, so the B should be 1, here only in the first one option, that is option number 2. Okay, now let's see the question number 23. For this we will see one diagram. It will help you in understanding. Like after the 1 month, heart will be forming, and in the second month limbs and the digits will be there. Okay, and at the 12th week or the first trimester, major organs will be developing. At the 5th month, first movement of the fetus, as well as the appearance of the hair on the head. And then on the 24th week or the second trimester, body is covered with the fine hair, eyelids separated, and eyelashes are formed. Okay, so that is what from where the question has come into this part. Okay, question number 123. Okay, so here they are saying that human pregnancy, the major organ are formed at the end of the 12th week. Absolutely correct, we have seen the, so A should be there. Okay, correct, they are asking, so A should be there. Second is like in human pregnancy, major organs are formed at the end of the 8th week. 8th week was not there. At what week major organs we have seen? At the 12th week, right? So at the 12th week, not the 8th week. Okay, so because of that, second should be, B should not be there. Okay, so B should not be there here also. Okay, so A is correct. C is saying that heart is formed after the 1 month of the gestation. Obviously after the 1 month of the gestation. Okay, that is correct. And D is saying in human pregnancy, limbs and the digit develop at the end of the second month. Okay, then human pregnancy, the appearance of the hair usually observed in the 5th month. Obviously just on the 5th month only we have seen the appearance of the hair on the body. Okay, so this is also correct. So A, C, D, E. A, C, D, E given in the fourth option, not in the first one. So we'll go with the fourth one. Next is, in the seeds of the cirrus, the outer covering of the endoplasm separate the embryo by a protein-rich layer. Protein-rich layer would be which one? That is the alluron layer. It is a simple question. We can mark it as an easy question. Then next question number 125 saying which of the following diagram is correct with regarding to the proximal and the distal, means P and the distal, proximal tubule and the convoluted tubule. So that same diagram given in our NCRT, so we can understand better from this diagram that this is the part of the PCT, right? So from the PCT, HCO3 was coming into the, means it is reabsorbing, right? So NaCl, water, nutrients, potassium ion, or these all are actually reabsorbing into the blood stream, and from the blood stream what is throwing out into the PCT, that is the H positive and H3 ammonia. Okay, that is going to the PCT part for the secretion. They are going for the secretion, right? So this would be for the secretion. And here for the reabsorption, what is the, that is the HCO3 from the DCT, HCO3, NaCl and H2O, that is from the, for the reabsorption into the blood stream. Okay, and what about for the secretion? So we can say for the secretion into the DCT, that is H positive ion, proton ion, these are for the secretion. Okay, these are for the secretion, and this is from the PCT part, this part would be for the DCT. Okay, so that is what they are asking in the question. So let us see in the diagram what they are asking. Here they are saying that for the reabsorption, that HCO3 okay, NaCl okay into the PCT, but into the PCT water is secreting, so it is incorrect. Okay, option number first would be incorrect. Okay, next is Okay, next is the here HCO3 is reabsorbing, water is reabsorbing, and salts are reabsorbing. Ammonia and the H positive of the proton ion are secreting it from the PCT, as well as from the DCT part. From the DCT part, NaCl reabsorption, water and HCO3, okay, and secretion for the proton and the potassium ion, that is secreting. So that is correctly defining here. But in the third one here, ammonia, okay, that is reabsorbing. These three are reabsorbing, and here ammonia and the H positive ion are secretion. This is correct. But into the DCT part, they are showing that NaCl is actually going for the secretion, so it cannot happen. Okay, and in the fourth one here again, water they are showing for the secretion. Okay, so it is again incorrect. So we'll go with the second option only. It is defining absolutely correct according to the NCERT diagram. Okay, now the question number 126, which many students was facing difficulty in this question. If you have studied very nicely to the diagram, as I always says that you have to read very nicely, you have to read very keenly. Okay, so from that only they have given, these are the rotors, right? These are the rotors from the center. So these rotors are for making the bubbles, okay, but the upper one most would be actually for the breaking the foams. Okay, so the foam creator are these, but the upper one would be the foam breaker. So this, if you have not read properly, then also you can logically solve it. Upper one would be foam breaker. Okay, so that is what they have asked into this question. So they are asking that bioreactor which is used as a foam breaker, from the given diagram, which part is for the foam breaker. So foam breaker is C only here, this topmost. Okay, so we'll go with the C. C is given in the fourth option, right? Now the next is question number 127, assertion reason based. Assertion is saying a typical unfertilized angiospermic embryo sac at the maturity is 8 nucleated and 7 celled. Okay, 8 nucleated and 7 cells. Okay, we have seen into this 3, 4, 5, 6, 7, 8. Okay, and 7 cells would be there. These two will be fusing. Okay, so nucleus would be 8 but cell would be fusing, so that is correct. And then we have seen in the assertion reason they are saying that egg apparatus has the 2 polar nuclei. Okay, so egg apparatus is having the polar nuclei, but actually we have not seen in the egg apparatus that 2 would be the polar nuclei. It is incorrect. So we'll go, reason is false, so assertion is true. So option number third one would be the correct answer. Next is the question number 128. A specialized membranous structure in a prokaryotic cell which help in the formation of the cell wall, DNA replication and the respiration is what? Okay, so here we can say they all are actually like cell wall formation, DNA replication as well as the respiration. That is what we have seen in the mesosomes. This was actually responsible for these all three functions, okay, in the bacteria cell. So I'll go with the first one. Then it is also again simple question only. Question number 129 is saying which of the following are the post-transcriptional events in an eukaryotic cell. Post-transcriptional, you can understand, like after the transcription, what was happening, like poly A tail was creating. Okay, poly A tail was creating, as well as poly A tail also known as the polyadenylation. Okay, so don't get confused with that. So I am writing here poly A tail or the polyadenylation, as well as the capping at the 5' end. Okay, so the capping at 5' end, this was happening at the 5' end and this was happening at the 3' end. Okay, so that is what they are asking here. Like transport of the pre-mRNA means not completely mature, means without capping and the tailing, to the cytoplasm prior to the splicing, means before splicing they are saying that it is transporting. It is not correct. Okay, incorrect. Why? Because without maturation, how the RNA can go out of the nucleus into the cytoplasm? So it is incorrect, and S should not be there. Okay, second is what they are asking, correct answer. Okay, and the removal of the introns and the joining of the exons. Okay, so removal of the introns would be there, and the joining of the exons means splicing and the slicing, they both are happening. Okay, so that is a part of the post transcriptional events. So this will happen, B should be there. Okay, so B is here and here. Okay, so two options eliminated. Third is the addition of the methyl group at the 5' end of the hnRNA. So that is what we have seen into the capping. Okay, then so C should be correct. Okay, and the addition of the adenine residue at the 3' end of the HN RNA. Okay, and base pair of the two complementary RNA. Two complementary RNA was not there; it was single strand, not two. So E should not be there. D should be there. So option number 2 only correct. Okay, the next question number 130. What is the pattern of the inheritance of the polygenic traits? Okay, so polygenic traits का pattern क्या होगा? Polygenic traits. Okay, what could be the polygenic trait? So simply you can say, like in the non-Mendelian inheritance pattern only we have seen the polygenic traits. Okay, so pattern of inheritance we have seen into the non-Mendelian only. So we'll go with the second option. The next question by 131 is saying which one of the following enzymes contains the heme in the as a prosthetic group. It is from the enzymology, right? It is from the enzymology. So what they are asking in this, heme group as a prosthetic group. So it is containing what? Heme group is actually containing as a catalase. Okay, so that is what we have seen. So simple question only. The next question number 132. 132 is each of the following characteristics represent a kingdom proposed by the Whittaker. Okay, arrange the following in increasing order of complexity of the body organization. So simply you can say like how to arrange them. Okay, so how to arrange them. Here the sequence is given here. Okay, so we have to find out the correct sequence or in the order. Okay, so that let's see. Okay, just a moment. Okay, so let's see what could be the correct sequence according to the Whittaker. So first of all, like hetero prokaryote. Prokaryote should be first. Okay, it should be the on the first. Okay, so prokaryote cell wall made up of the polysaccharide and the amino acid, that is correct, and it is arranged as a prokaryotic cell, monocytes. Okay, and then eukaryotic, and the eukaryotic with the cellular autotrophs with the cellular body organization. Yes, it should be the second one. Okay, and then heterotroph, no, then multicellular should be there, so C3 should be there. Okay, so 1 2 3 I mean CEA. Let's see CEA, and then DB should become CEA given here, CEA given here. Let's see DB or the BD. Okay, okay. Okay, so eukaryotic autotrophs with the tissue organs, and here they are saying tissue organ and then organ system, like heterotroph. First eukaryote and then heterotroph. Okay, so first D and then E. So the CEA, then DB. Okay, CEA DB. Okay, CEA DB in the second option. Okay, so given here correctly. Now let's see that question number 133, memory based question only, father of the ecology they are asking. If you have memorized, then only you can give the answer. It was not a typical, but if you have read in India, if you they are saying, so it is Mishra, common only. Okay, now let's see question number 134. So Alfred Wallace. Again, see, I was telling that scientist name you have to read. It is very important if you are reading the scientist name and their experiments, so it would be helpful. So here the question is in front of you related to that topic which I told. Okay, so the Hershey and Chase experiment we have seen in which, let us match with this perfect. Here question is in front of you on 34. So Alfred Wallace, so means A, loosely packed or the light stain. Okay, or either a DNA is a genetic material. Okay, so we can say it is DNA is genetic material. It would be matched with the fourth one only, not with the light one. Okay, euchromatin could be the, yes, B3 should be there. Okay, B3 only in the one option and A4 here. Okay, so we'll go with the third option because B3 given only in the one option. Okay, so here completely matched. Next is question number 135. Neoplastic characteristic of cells refers to what? Neoplastic characteristic they are asking for what conditions given below. Okay, so that is actually a mass of the proliferating cells. Okay, proliferating means it will give rise to the complete development and then give rise to the new cells related to the same cells, like X is growing and then growing into X1 and X2, right? It is X1 and X2, so it is proliferating, right? It is proliferating from one to the another. Two differentiation is there, but it is proliferating from one to the another cell. So mass of the proliferation, that is what indicating the neoplastic characteristic of the cell. So A should be there. Okay, and then rapid growth of the cell. If the growth is there, then only the condition would be increasing now. So B also should be there. Okay, and then invasion and the damage to the surrounding tissues, that we can say it is a basic invasion means to here are the growing cells. If any cells is particularly damaged because of some reason, because of some damaged protein, so it will show the detachment from the nearby cell, it will go to the another healthy cell area, and then it will make itself to attach to the nearby healthy cell and invade itself into this area and perfectly fit and then go. Okay, so that is what the invasion. And damage the surrounding tissues means, and then it will start spreading its damaged protein into the nearby cell, and the nearby cell also will become damaged, right? So this is also correct, it is a property of the neoplastic cells. So A, B, C, all three should be there here. And those confined to the original location, it is not the original location, it will migrate from one to the another cell, so we can say it is incorrect. So A, B, C only. Okay, now let's see the next question, number 136. Question number 136 is that statement based again. DNA fragment extracted from the gel electrophoresis can be used in construction of the recombinant DNA. So in the recombinant DNA fragment we are seeing that it is we are using it. So DNA fragment only we are doing with the help of the restriction enzyme, and then on the gel electrophoresis by using the agarose gel on the agarose gel plate, and then we are making it by the recombination of it and then making the fragment to move. Okay, so that is what we have seen in the gel electrophoresis. And the smaller size of the fragment are observed near to the anode, while the larger size fragment are near to the valves of the agarose gel. How to understand? Let's see. Suppose this is the agarose gel plate. In this there would be the valves. Okay, these are the valves, and we are having the fragment of the DNA, and we know the fragment of the DNA is negatively charged. So here this side negative charge, this side positive charge, cathode and the anode we have added. Okay, so here anode is there and here is the cathode. So whenever the smaller size of the fragment is there, so definitely the DNA is negative charge, it will start moving very fastly towards the positive end. So smallest fragment, that means the largest the distance, and biggest the fragment, biggest not in the size, it would be bigger according to the molecular weight. Okay, so the biggest the fragment, it would be near to the cathode. Okay, so that is what they are telling in this question. That is like smaller DNA fragment are observed near the anode. Okay, smaller is going down towards the anode, and the largest fragment are found near the well in the agarose gel. Near the well in the sense means towards the cathode, or heavy people, like if any heavy or the fatty person will start racing. Okay, so in that racing, who will be win? Those who will be very slim and not having the body weight, so that person will win the race. And those who are having very heavy weight, so they will not win the race, even they will be towards the pole from where they have started. Okay, so they both are correct, that is what they are telling in this. Okay, next is question number 137 here, that adenosine, adenolic acid, adenine and the alanine. Alanine is a amino acid, simplest, second smallest amino acid is the alanine. So D4 given here, and then adenolic acid is nucleic acid. So nucleic acid means here nucleotides we should say. So B2, so B2 is here. Okay, so B2 and D4, option number third would be there, and A3 let us match. A3 adenosine is a nucleoside. Okay, A3, that is correct. Next is question number 138. 138 is here. The reduction division of the human female gametogenesis start earlier than that of the male gametogenesis. Obviously from in the females we have seen at the embryonic stage, right? But into the males it is starting at a puberty. But in the males it is started at puberty, right? So that is a basic difference between the male and the female, so which we have seen. So this is true. Okay, the first one is true. Second is the gap between the first meiotic division and the second meiotic division is much shorter for the males compared to the female. Like into the males there is not even the shorter they are saying, but actually there would be not too much difference. So at one particular stage only it is completing. Okay, and in the females too much gap is there. Okay, so this is also true, not absolutely, but let's see other options. Okay, third is first polar body is associated with the formation of the primary oocyte. First polar body because of the secondary oocyte, not because of the primary oocyte. So this option will eliminate. So C should not be there. Okay, and A should be there. So option is here in front of us. Let us see the others. that LH surge lead to the disintegration of the endometrium. LH surge was actually causing the ovulation. Okay, it was not the LH, it was caused the ovulation, not the endometrium disintegration. It was because of the progesterone low level as well as the FSH level, not because of the LH. So it is also incorrect. So A and B only. Okay, now let's see the next question. All the living members of the class cyclostoma. Class cyclostoma is having the ectoparasite, circular mouth, right? It was the circular mouth, jawless fishes of the agnatha. We have seen a agnatha. Okay, so they all was ectoparasite living on the some other organism surface. Okay, next is the question number 140, that is asking for the assertion reason based again. So let's see what it is saying. The primary function of the Golgi apparatus is to package the material made by the endoplasmic reticulum and deliver it to the intercellular target and outside the cells. Outside the cells it is releasing out. Absolutely exact complete function of the Golgi body they have given as per of the words of your NCRT, because the first function they are telling for the Golgi body is to package the material which is made up in the endoplasmic reticulum and then deliver it to the intercellular target and outside the cell. That is absolutely correct. And second is saying that vesicles containing the material made by the endoplasmic reticulum, it will fuse with the cis phase of the Golgi body and they are modified and released to the trans phase of the Golgi apparatus. So we have seen like cis phase and the trans phase, right? So here and here it is going outside cis phase and then going via the trans phase. So that is what into the Golgi apparatus we have seen, right? So on the basis of that they are saying like it is moving from the cis to the trans phase and then going out of the cell, right? So that is of the that is what we have seen into the Golgi body function. So let's see is it explaining or not, because in the first in the assertion they are saying that material is packaging and then delivering. How it is delivering? By making the fusion with the cis phase and then releasing out via the trans phase. So that is what they are explaining. First they are telling the function is packaging and the delivering by this process. That is absolutely correct. Okay, so this we can say it is the correct explanation also. I hope this would be clear. Here don't get confused, it is given as per your NCRT as well as we knows the basic function. Okay, so that is what they are telling in this one. Okay, now let's see the question number 141. Scutellum, non-albuminous seeds, then epiblast, then the perisperm. Okay, so let's see. Scutellum is what? Scutellum is into the groundnut A3. Okay, not in the groundnut. Sorry. It is not into the groundnut. It was actually into the cotyledonous. Okay, scutellum into the cotyledon. Okay, of the seeds. So in the seeds only. So A should be 2. Okay, so A2 given here, first and the fourth. So these two eliminated. Now let's see B is non-albuminous seeds. So non-albuminous seeds was the groundnut. So B should be 3. Okay, B3 A2. Okay. Answer is here. Okay, next is question number 142. 142 again assertion reason based. See, many times they have given assertion reason based, and it was expected. This question was expected. So all the vertebrates are the chordate. Yes, because in the phylum Chordata, we have seen like some was the urochordate, some was cephalochordate, and some was vertebrate, right? Some was vertebrates. And in the vertebrate we have seen some was having the jaws and some was not having the jaws. So agnatha and maybe the gnatha. So that is what they are asking in this question. So all the vertebrate are the chordate. Yes, all the vertebrates are actually the part of the chordate phylum. Okay, and all chordates are not vertebrate, because some may be the urochordate, some may be the cephalo, and some may be the vertebrate. So that is absolutely correct. And then the member of the subphylum Vertebrata possesses the notochord during the embryonic period. In the vertebrates only we have seen at the embryonic stage they are having the notochord, and the notochord is replaced by the cartilaginous or the bony vertebral column in the adults. Okay, so that is what correct only, and because of that only they all are vertebrates and not all the chordates are actually the vertebrates. So that is what they are telling and explaining also. So we can say it is correct explanation and both are correct. Okay, Next is question number 143. Identify the incorrect one, not correct. Each antibody, here the diagram I have attached here, actually this part is N terminus where the antigen binding site is available, and this is the FC part. This part is actually FC part. Okay, and then above part is FAB means fraction in which the antibody will interact, fraction in which the constant region is there. So in the constant region there would be like disulfide bond present between the two heavy and the light chain, and in the FAB part there would be the antigen binding sites. Okay, so this part is actually the N terminus. Okay, and this part is C terminus. So you have to be very careful from the diagram given in the NCERT. Okay, so here each antibody has a two light chain and the two heavy chain. Okay, each heavy chain, heavy and the light chain are held together by the disulfide one, obviously because of that only they are tightly binding, and the antigen binding site is located at the C terminus. It was actually N terminus. Okay, so because of that it would be incorrect, so it would be our answer. Constant region of the heavy and the light chain are located at the C terminus. That is also correct, so we'll go with the third option. The next is the silencing of the silencing of a specific mRNA is possible by the RNAi because of what? Okay, so because of what it was? Because making it self as a complementary double stranded. It was a simple question. So 144 first option. Then next question number 145. Gene R and Y follow the independent assortment if the RRYY produce a yellow round seed and the RRYY produce the wrinkled gene seeds, what will be the phenotypic ratio of the F2 generation? For this you have to solve it by making like dihybrid cross. It was obviously it was a 9331 ratio in the dihybrid cross, and it was a phenotypic ratio, not the genotypic. Genotypic ratio was 3:1, right? It was 3:1 in the F1 generation. Okay, in the F1 generation we have seen it was 3:1. So we'll go with the third option. The next is histones are enriched with what? Histones are enriched into the lysine and the arginine because they both are positively charged, and our DNA is a negative charge. DNA is negative charge. So because of the negative and the positive, they both are interacting with each other, and the histone will make a tightly binding of the DNA on itself. Okay. The next question number 147, the first menstrual cycle, again very simple halva question, Minar. Okay, then 148, let's see, heart, kidney and GI tract and adrenal cortex. Simple question only. Erythropoietin was from the kidney, heart was secreting A and F. Simple question. Heart was secreting A and F. A3 should be there. A3 given only in the one option. Answer is here. B2 kidney, aldosterone. Okay, and gastrointestinal tract, like from the GI it was secreting, and adrenal cortex aldosterone, sorry, it was aldosterone, and from the kidney erythropoietin. Okay, it was doing the erythropoiesis. It was doing the erythropoiesis. Okay, so the fourth option. Next is question number 149. Here the protein portion of the enzyme is called as it. Protein portion of the enzyme is obviously apoenzyme. Alone enzyme is apoenzyme. If the cofactor is attached, then only it would be the holoenzyme. Okay, it was the holoenzyme if the protein as well as the enzyme is attached over it. Okay, now let's see the question number 50, that is which of the following is a unit of the productivity in the ecosystem. Simple question only. It was a KCALM and here inverse. Okay, question number 150, it would be option number 4. If you have seen the ecosystem ecology chapter, so it would be quite easy for you. The next question number 151, sweet potato and the potato. Sweet potato, we know potato is actually the stem, right? Potato is the stem, modified stem, and the sweet potato was a modified root, and they both was the example for the analogous. They both was the example for the analogous. If it is analogous, so definitely convergent evolution. Okay, analogous with the convergent evolution. So we'll go with the analogous with the convergent evolution. The next is 152. With the help of the given diagram, we have to find out child has no disease and the carrier. One child should be no disease, means normal, no affected, and one should be the carrier. What is the percentage they are asking? So let us see, let us focus on one thing, that is this family. This family we have to focus. So first what they are saying, let us see, X it is a female and For the father, XY, father is disease, so I am showing here. Mother is disease carrier, so here. So let us see what could be the progeny. So X with the X dash. Okay, so here X with the X dash, and here this X with the X dash. Okay, then Y and Y. Okay, so let us see with the X dash and X. Okay, so this would be the diseased, this would be the diseased. Okay, it would be the carrier, it would be the carrier. Here I wrote it wrong, it should be normal. Okay, normal X. Okay, it should be the normal X, normal X. Okay, so here this would be the disease, this would be the carrier. Okay, so because of this one, like from the 4, 1 is carrier and 1 is only the disease. So 1 by 4 is a carrier, 1 by 4 is a disease, and 2 by 4 are the normal. 2 by 4 would be the normal conditions. Okay, so let's just see what they are saying here. So 1/4, obviously it is affected. 2 would be the normal, 2 would be the carrier. Okay, so I hope this would be clear to you. Question number 153, let us see. Assertion reason based again. Lot of the question are there related to the assertion reason based. Okay, question number 153 here is saying that cells of the tapetum possesses the dense cytoplasm and generally have more than one nucleus. So dense cytoplasm as well as more than one nucleus we have found into the tapetum. Absolutely correct. And presence of more than one nucleus into the tapetum, it will increase the efficiency of nourishing the development microspore mother cell. Was it correct? Absolutely wrong. So because of that reason should be false and assertion is true. Okay, so it would be the third option. The next, how many meiotic division, meiotic and mitotic division you need to occur in the development of the mature female gametophyte from the megaspore mother cell in the angiosperm plant. Okay, so how many divisions was there? Actually it was because of mitosis was 3. Okay, mitosis was 3 and meiosis was only 1. Meiosis was only 1, then only the complete division was occurring in that. So meiosis 1, mitosis 3. Okay, third option. Okay, the next question number 155, which of the following is the example of the zygomorphic flower? So for that I hope you made a code word for this type of the question. So it was a P in which the zygomorphic flower was there. Petunia, datura and chili was not showing the zygomorphic, it was the actinomorphic. Okay, next question number 156. After the maturation, primary lymphoid organs, the lymphocytes, migrate for the integration with the antigen to the secondary lymphoid organ or the tissues like. So in the bone marrow and the thymus, right? Bone marrow and thymus. Okay, so they was actually the primary lymphoid organ and rest other are the secondary lymphoid organ. Okay, so these two are the primary lymphoid organ, rest others are secondary lymphoid organ. So C, D and E, option number 4. The next question number 157 here. Fig fruit is a non-vegetarian. Yes, why it is non-vegetarian? Most students are asking me that why it is non-vegetarian, because in the fig fruit the wasp is laying its egg. So like we are human, okay, and we are inhaling the chicken or mutton, so in that case we became the non-vegetarian. So fig fruit is getting the wasp egg, so in that case it is also like non-vegetarian. So that is okay, it is a non-vegetarian as it has enclosed the fig wasp into it. Okay, because of that it would be non-vegetarian. So this is correct. But here they are saying like in this case, okay, it is a non-vegetarian, but as it has enclosed a fig wasp, not the fig wasp, wasp egg. So because of this it is incorrect, not because of the non-vegetarian, because of the fig wasp, it was because of the wasp egg. Okay, so because of this statement would become wrong, not because of the non-vegetarian. So students was getting confused that it is non-vegetarian, because of this it is incorrect. It was non-vegetarian only, but not getting the egg of the fig wasp egg. Okay, so it is incorrect. Second statement is A fig wasp and the fig tree exhibit the mutual relationship as the fig wasp completes its life cycle in the fig fruit, and the fig fruit get pollinated by the fig wasp. That what we have seen, it was incorrect only, na? Because fig wasp was first of all not there, and fig tree exhibit the mutual relationship. Okay, like wasp and the fig tree was showing the mutual relationship. Because of that, it is incorrect. As the fig wasp completes its life cycle in the fig fruit, and the fig fruit get pollinated by the fig wasp. No, incorrect. Vice versa, they are telling. Okay, so it is also incorrect. So we can say that both are incorrect, both 1 and 2. Second option should be there. Fig wasp and the fig tree get exhibit the mutual relationship. Fig tree and the fig wasp, no, wasp egg, which should be incorrect only. Okay, it is incorrect. Okay, then question number 158, what is the main function of the spindle fibers during the mitosis? Spindle fiber function is to we can say separate the chromosome, not to synthesize the DNA cell growth. No, first option only. Which of the following is the characteristic feature of the gymnosperm? Gymno means naked. Gymno means naked seed. Okay, naked seed. Okay, so let's see what could be explaining seed are naked. Okay, so second option only. Then next is consider the following statement regarding the function of the adrenal medullary hormone. Adrenal medullary hormone, here it is showing in the first one pupillary constriction. It should be pupillary dilation, not the constriction. At the time of the high blood pressure, it will show the dilation here. So in the dim light, it would be dilated. Here in the in the bright light, it is contracting. Whenever we are having the panic attack, in that situation, our pupils will become dilate. Okay, so here because of this, it would be incorrect. Okay, so the A should not be there. Let's see, hyperglycemic. Hyper means more, glycemic means glycogen will break, so definitely glucose level will be increasing. More glucose means more glycogen is breaking, so that means the more glucose will be there. So B should be correct. And piloerection, obviously we have seen, and the increase the strength of the heart contraction, obviously. So B, C, D all should be there. B, C, D 3. Sorry, second option. Okay, next question number 161 is why can't insulin given orally to the diabetic patient? Because it could be elicit immunity. It could be digest in the gastrointestinal tract. Simple question only. We have seen many times to this question. The next is bryophyta, pteridophyta, gymnosperm, and the angiosperm. So let us see. So what it is saying, like 162, we have to find out the correct matching for the example. So I hope in this time period, in this few seconds, you have matched it very nicely, because it is simple question only. Pteridophyta is what? Pteridophyta is actually the polytrichum. So A should be 3. No, sorry, Salvinia. It is not polytrichum, it should be Salvinia. So let me correct it. It should be the Salvinia. So A is 4. Okay, A4. A4 here, A4 here. Okay, let's see B. So these two eliminated. So B is what? B is the bryophyta. Okay, bryophyta is polytrichum. So B3. B3 here. B3 here. Second. So second option. Okay. Next is 163. Who proposed the genetic code for the amino acid should be made up of the 3 nucleotide. Again, memory based question here. That was by the George Gamow. If you have learned it, then only you can solve it. Okay. The next question number 164 is Evil Quarter, Ex-situ conservation, then Lantana camera, then Dodo. Dodo, Mauritius, and extinction, going to extinct. Okay, so simply D4. Okay, D4 should be there. Okay, D4 means these two eliminated. Okay, and A3 again given in the same. B, we can match with the B. Ex-situ in the sense like ex-situ conservation by the cryopreservation. Okay. What could be causes of the biodiversity extinction? हो गया है. Okay, it would be cryopreservation. Then Lantana would be okay. So B should be 1 only. So B is here 1 and C 2. Okay, so we'll go with the second option. Okay, next is 160. Which of the following hormone released from the pituitary is actually synthesized in the hypothalamus? Synthesized in the hypothalamus means hypophyseal portal system, which was giving the signal to the body, right? In the hypophyseal portal system, hypophyseal portal system, it was coming from the hypothalamus, from the hypothalamus to the anterior pituitary, right? From here to the anterior pituitary. But from the hypothalamus, neurons are coming directly to the posterior. It was not giving to the anterior pituitary, but via the hypophyseal portal system it was giving the signal to the anterior pituitary, and it was synthesizing and secreting into the bloodstream. But from the hypothalamus, direct neurons was coming and synthesizing the hormone into the posterior pituitary, and whenever required it will release. So this is also the condition that into the posterior pituitary it is having the ability to store the hormone. Okay, so let us see. ADH is only the part of the posterior pituitary, and this is again anterior pituitary. This is the anterior pituitary. This is the anterior pituitary. Okay, so these things. So we'll go with the second option. Okay, next is question number 166. Here the role of the water vascular system in the echinoderm. Water vascular system into the echinoderm we have seen for the respiration and locomotion, okay, but not for excretion, digestion, capture and transport, okay. A and C should be there. A and C given in the option number 2. The next question number 167. That is, which of the following type of the immunity is present at the time of the birth and is non-specific type of the defense in the human body? At the birth, what was present? It was innate immunity. Simple question. Then into the bryophytes, the gyma will help in which of the following? Gyma was actually doing the asexual reproduction. Simple. Again simple. The next question number 169, in the frog, renal portal system, okay. Specialized venous connection heart that connect what? Obviously not intestine and the liver, not to the kidney, okay. Kidney it is there, but not the intestine and the liver. Kidney should be there and the lower part, okay, not the intestine and the kidney. From kidney to the lower part of the body, not only the intestine, it should be the lower part including the intestine. Lower part means it is including the intestine as well. Okay, so we'll choose the option number 4. Don't get confused in this, because intestine is a part of the lower body parts, na? So I will say that kidney and the lower part of the body. So we'll go with the option number 4. The next question number 170 statement. In ecosystem there is a unidirectional flow of the energy of the sun from the producer to the consumer. From the producer to the consumer only we have seen the flow, from the producer, then the consumer, then secondary consumer and all. Okay, primary consumer, secondary consumer, tertiary consumer, like that only we have seen. So that is what they are asking. Like first source is from the producer and it is getting the energy from the sun. First source is the sun for the producer. So that is correct. And second is ecosystem is exempted from the second law of thermodynamics. Second law of thermodynamics was telling about the disorderness. Okay, so in that case, like orderness and the disorderness, if we know in the thermodynamics, in the physics, as well as into the enzymology of the biomolecule chapter in zoology, if you have seen that, so you can understand it. So it was not the second law. Okay, so it was not the second law. It is in the proper order, and this second law is explaining for the disorderness, right? Second law was disorderness. Okay, and this is in the order, right? This is in the order, so because of that it would be incorrect. So statement 1 is correct and 2 is incorrect. We'll go with the third option. Okay, next is question number 171. Which of the following statement about the Rubisco is true? For the Rubisco we have to find out which one would be correct. Okay, let's see which one could be the correct. Like it is active only in the dark, no. Highly affinity for the oxygen than the CO2, no, it was actually getting the CO2. And the enzyme involved in the proteolysis, no, not in the proteolysis of the water, it is the carboxylation of the RUBP, absolutely correct. We'll go with the fourth option. Then next 172 is, which of the following enzyme is not essential for the gene cloning? For the gene cloning, restriction enzyme is very important, yani ligase is very important, recombinase, polymerase and mutase. If recombinase is there, okay, mutase, first of all mutase means it will cause a mutation. We have not seen the mutation at all, it could not be there. And polymerase, like recombinase, polymerase is extending the complete distance, so it should be available, not like it should not be there. It should be available, then only the polymerization would be there, right? So recombinase was not there, recombination was not happening in that stage. Okay, so the D means it should not be there. So C and D should not be there. What that is what they are asking, should not be present. So C and the D. So first, the next question number 173 here, parthenocarpy. Achha, what we have to find out? Correct statement. Okay, so here correct statement means parthenocarpy can be induced by the auxin. Okay, by the auxin only parthenocarpy they are asking. Okay, that is correct. And plant growth regulator can be involved in the promotion as well as the inhibition. Plant growth was doing the like if it is working more, it is regulating, like for example abscisic acid here. So let's see here option also they have given for the abscisic acid, which I was telling. Abscisic acid is a plant growth promoter. It was not the promoter, it was the inhibitor. So D should not be there. Okay, D should not be there, right? And A and B both are correct. So here answer, and it is first of all it is the inhibitor, not the promoter. Okay, so it is incorrect. So A and B okay, and C okay. Differentiation of the prerequisite for the redifferentiation. Okay, first of all prerequisite condition for the redifferentiation, then only the dedifferentiation would be the first differentiate and then again redifferentiate. So first of all differentiation, then dedifferentiation, and then redifferentiation would be there. So that is also correct. So A, B, C, all the three are correct. The next question number 174, like which factor is important for the termination? Again simple halva type question. Okay, so the halva type of question means termination. For the termination, what was important? It was a row factor, row dependent and row independent. Row dependent as well as row independent. Okay, so these two process only we have seen, right? Now let's see question number 175, that is frog respire in the water by the skin and buccal cavity, on the land by the skin and buccal cavity on the and the lung. So on the land we have seen by the 3 mode, and underwater only by skin. Okay, underwater only by skin, but on land we have seen by the 3 mode, skin, buccal cavity as well as by the lungs. Okay, so on the land it is correct, but for the water they are saying by skin and the buccal cavity. For the water it is not correct. Okay, let's see. They are saying statement true is true for the water. No, it is not true for the water. It is wrong for the water. Statement is true for both, then no, it is true, false for the water but true for the land. Okay, that is correct. Okay, so next 176 is twins are born in a family that lives next to your door to you, and twins are a baby boy or a girl. Which of the following must be true? If they are see how to understand this type of the question. Here they are saying monozygotic twins could not be there, because like they are the twins, maybe one boy and the one girl, so not the monozygotic, it would be the fraternal twins, simple only. Not the monozygotic, it would be the dizygotic could be there, and it is the fraternal. Okay, so it should not be the second correct. Conceive through the in vitro, that how we can say conceiving through in vitro or not. At 75% genetic, no, it is not there. So we'll go with the second option, which is best, that is the second. The next question is again from the botany. Which of the following is the microbes? Microbes is not involved in the preparation of the household product, which is not involved into the household. This trichoderma, we have seen trichoderma polysporum into the cyclosporin B. Okay, that is immunosuppressant. Okay, so that we knows very well. Okay, it is not like using in that. So C should be there. Okay, because they are asking which is not involved, so C should be there. And here, and Aspergillus niger is okay, again it is also not using into the household, so A and C should be there. Okay, in one option only, second option. Then next is 178, again human reproduction. Progesterone is from the corpus luteum, simple one, A4. Okay, catecholamine is what from the adrenal medullary hormone, D3. And melanocyte stimulating hormone, pars intermedia, C1. And relaxin, C1, and relaxing from the ovary. Okay, so what is relaxin B? Okay, so A should be progesterone A4. Okay, so A4 here, and relaxin catecholamine D3 we should match, D3 A4 D3 here, option number first. Okay, 178 first option. Then next question number 179. Here is screening test they have given. Acha, one thing I should tell you first, then you will understand the question. Simple only. But if we know the experimental base, suppose this is like plasmid, any plasmid, any vector. Okay, in this we are having a gene, that is a LEXZ gene. It is already here. LEXZ gene is present, which is responsible for the beta galactosidase, and this beta galactosidase is able to interact with the X-gal. If it is present in the medium, if on the plate we are having a media, and on that media we have poured or we have included some X-gal. Okay, it is what X-galactose, which is a pigmented. Okay, so if this plasmid, first we will insert it into the bacterial cell, means transformation, and it is already recombinant. So it will go into the, on the media, it will grow and it will interact, the X-gal will interact with the X-gal, and the beta galactosidase will interact. It will give the blue colored colonies. It will give the blue colored colonies of the, after the growth. Okay, so the blue colonies is indicating that the gene is actually present, available. If suppose we have inserted our gene of interest into this particular area, our, this is our gene of interest, so because of this LEXZ gene will disrupt, no beta galactosidase will be there. No beta galactosidase means no interaction with the X-gal, so no blue color colonies. What will come? Colorless colonies will be there. Colorless colonies, right? So colorless colonies indicating, colorless means white colonies. Okay, so that is what they are telling here in this question. So blue and white selectable marker has been developed, which differentiate the recombinant colonies from the non-recombinant colony on the basis of their ability to produce color in the presence of a chromogenic substrate. So two statement they have given. Statement one is blue colored colonies have DNA inserted in the plasmid. Blue color colonies was showing inserted, or the white color was showing. Okay, so it is incorrect. White color colonies means there was insertion. So insertion and activation will cause the white color colonies because of no beta galactosidase. No beta galactosidase interaction with the X galactose. So because of that it is incorrect. Second is colonies without blue color, without blue color have the DNA insert in the plasmid and are identified as a recombinant colony. Absolutely correct. That is what indicating. So fourth option. Okay, so here again simple question, which one of the following equation represent the Wall-Hurst logistic growth of the population? That complete diagram, formula based question, simple. dn upon dt is equal to r and k minus n upon k. Okay, so second option. So simple question only this was. So I hope these all question will help you in understanding the NEET 2025, and I hope you will get the good marks. And don't panic that you are not getting the rank, or you will get or not. You will get if you are having the marks. You can expect. It's not, I am not giving the assurance that you will get or not, but I can expect that if you are getting 500 plus marks, definitely you will get the C. Okay, so just don't panic. If the question paper was hard, so like cutoff will be down. Okay, so we can expect like that in the NEET 2025. Okay, so let's hope for the positive. Stay positive, stay blessed. Okay, take care, and thank you.